Hello everyone and welcome to my YouTube channel. So in our last lesson we have started the discussion about the DNA extraction and purification. Okay, and there we have seen that there are different methods available for the isolation of the DNA and for further purification. So from today's class onwards we are going to discuss each and every method in detail, starting with the first one, which is a very basic and general method utilized in the laboratories, and that method is the phenol chloroform DNA extraction method, which is a kind of organic DNA extraction method. Remember, we have read that there are organic DNA extraction method, then we have enzymatic DNA extraction method and inorganic DNA extraction method, and certain physical DNA extraction methods are there. So already we have get that overview on our last lesson. So this phenol chloroform DNA extraction method is a kind of organic DNA extraction method, and this is based on the principle of liquid extraction method. Okay, I have told you that what this liquid extraction method is. So today we are going to discuss it in details. So starting with the topic. Okay, so before starting this topic, we would be looking forward for certain points, and we should remember these points while we are studying this lesson. So the first point we should keep in our mind is about the principle of this method. Okay, if you don't know the principle on which this method is based on, any study you conduct and you don't know the principle, that is a waste, total waste. Okay, so first of all you should understand the principle of the study. In this case you should know the principle of phenol chloroform DNA extraction method, that how are we going to isolate the different components. Okay, then what are the different chemicals we are using in this particular extraction method, what is the basic role or function of those chemicals. So every function of each and every chemical we are utilizing here, you should know that what function it is performing in this particular extraction method. Then the methodology, the steps involved in this method. So we will be discussing that all the steps which are involved in this DNA extraction method till the point we get our DNA. Okay, after we get the DNA, we need to precipitate it because it is present in the solution, right? We need to precipitate the DNA. There are different methods by which we can precipitate the DNA. We will be discussing them too. Then the DNA quantification. How can we measure the amount of DNA that we have isolated? So each and every point written here, at the end of the lesson, you should be able to answer all these questions, all these points. So study like that. Okay, so starting with the basic principle first of all, it's right here, that is the phenol chloroform DNA extraction method is based on the principle of liquid extraction method, right? We have already discussed. But what this liquid extraction means? So I have already told you that different molecules and different biomolecules in this case, they have different solubilities in different solutions or solvents. And depending upon this solubility of any component or the biomolecule into various solvents, we can isolate them, we can separate them. Okay, so this is the only principle of this liquid extraction. So according to this liquid extraction, various molecules or compounds, they get separated on the basis of their relative solubilities in two different immiscible liquids, right? Usually those immiscible liquids are your polar and non-polar solvents. So we are using two different immiscible liquids. Those liquids are generally polar and non-polar solvents. generally water and any organic solvent you can take in case of non-polar, and those solvents are utilized for the separation of different biomolecules on the basis of the solubilities of the biomolecules in either of that solvent, right? So the separation of the DNA molecule occurs on the basis of its solubility in the immiscible solution. So this is what the principle of your DNA extraction method based upon the phenol chloroform. Okay. Then what are the different chemicals we generally use? So the first chemical we use is tris, right? Then comes to the EDTA. Then we have SDS, that is sodium deutesyl sulfate. Then certain salts we use, that is your sodium chloride and magnesium chloride. In case of sodium chloride, you can also use sodium acetate. We need a salt. We will discuss that why we need this salt. After that, we require phenol, chloroform and isoamyl alcohol. So these are the various chemicals we use in this extraction method. So these chemicals are basically used in case of your DNA extraction from different cells like your blood cells, or you can use these chemicals from the extraction for the extraction of DNA from bacterial cells. So these chemicals can be used in that. For plant DNA extraction, I'll be explaining you what other chemicals you can use, because we have a rigid cell wall there and we need to break that cell wall. So for that we require certain other chemicals. Okay, so we will be discussing about them too later on. But for now, if we are utilizing the blood cells and the bacterial cell, these chemicals are enough. So now we will be discussing about each and every function of these chemicals that we are using. So the first chemical here is the tris. Why we are using tris? Because tris here is providing a particular pH to the solution. Okay, DNA require the pH. Not only DNA, every component in our body, it functions at a particular pH. If we remove that component from that pH, that won't function properly. The function will be disabled. So we need to provide a particular pH to the component so that it won't get degraded. So for providing a particular pH to that component, to the DNA, we require this tris. Because what is the function of tris here? It maintains the pH of the solution, which is around 7.6. Okay, so the whole procedure needs that pH only. So this tris will help to maintain the pH of the solution throughout the reaction. Then second function of this tris is it interacts with the lipopolysaccharides of the cell membrane. So when it interacts with the lipopolysaccharides of the cell membrane, which is the basic structural component of the cell membrane, this tris will make the cell membrane permeable. And when the cell membrane becomes permeable, it will be lysed soon. Okay, so it helps in the lysis of the cell membrane. Then we have EDTA. So EDTA, the role of EDTA is what? Chelation, right? So it chelates the magnesium ions which are present in the solution. Now why in the solution? Because when lysis occurs, so what happens, this EDTA generally helps in the lysis, because we know that major enzymes, and certainly if we talk about the lipopolysaccharide and the structural envelope of the cell membrane, it requires the magnesium ions for that structural integrity, moreover for the enzymes too, for proper functioning of the enzymes, because certain metals like magnesium and calcium, they work as a cofactor for various enzymes to work properly for their catalysis, right? These magnesium or calcium ions, they bind with the active site of the any enzyme and make it function properly. Okay, so the basic role of this EDTA is to remove those ions, those magnesium ions from the from those components, from those enzymes, so that those enzymes will not function properly. And in this case, we need to degrade or we need to block an enzyme that is known as DNase, so that our DNA won't get degraded. Okay, so the basic function of EDTA is to chelate the magnesium ions, which is working as a cofactor for the enzyme DNase. And DNase, if we want to block the DNase, we need to chelate those magnesium ions so that this DNase won't degrade or destroy your DNA. Okay, so EDTA is the chelating agent which chelates the magnesium ions, which act as a cofactor for the enzyme DNase, which is an enzyme that lyses the DNA. Hence, it blocks the DNase activity. Along with it, it also helps in the lysis of your cell envelope. Then comes the SDS. So SDS is a kind of anionic detergent, and this SDS helps in the lysis by removing the lipid molecules. So it helps in removing the lipid molecules, and so resulting in the disruption of the cell membrane, and also it helps in the denaturation of certain membrane proteins, right? So we need these components for the lysis. We can use this component separately one by one in a stepwise manner, and also we can prepare a lysis buffer, which are generally used in the kits method nowadays. So in various kits you will find the lysis buffer, and those lysis buffer carry these reagents, carry these chemicals. Okay, so you can use them separately, or you can prepare a lysis buffer with the help of these chemicals, and you can use them further. Then we have the salts, that is the sodium chloride or sodium acetate. So what happens when we use these salts? This is generally for the precipitation. We will be discussing that we can use these salts, but for protecting the DNA during the time of lysis or after lysis, we use these salts so that we can preserve or protect the DNA. Because when all the cell debris is present in the solution, the DNA has a chance to get mixed with that debris and will be removed. So to maintain the structural integrity of the DNA, to protect the DNA from mixing, we use certain salts. One of the salt is sodium chloride and the other one is the magnesium chloride. So sodium chloride, what function it generally performs? It has a positively charged sodium ion on it, right? So what it will do, the positively charged sodium ions, it forms the ionic bond with the negatively charged phosphate ion that is present on the DNA. And when this ionic bond is formed, this bond will neutralize the DNA, because the positively charged sodium ion will neutralize the negatively charged phosphate ion of the DNA, which helps the DNA to come together. Means it's kind of like they come together, make a clog-like structure, and that will be protected from denaturation. So this is the role of NaCl there. Then we have magnesium chloride. So magnesium chloride, what function it plays? It protects the DNA from mixing with other organelles, as it blocks the negative charge of the lipoprotein of the cell membrane. So the lipoprotein which is present on the cell membrane, it has a negative charge. So the magnesium ions, so the magnesium ions, they generally form the bonds with the negatively charged lipoproteins, and further protects the DNA from getting mixed with that debris or the other organelles present there. Then we will be using this phenol. What is the function of phenol? Phenol, when we add phenol in the solution, it has a particular role in denaturing the proteins. So it will denature the protein and leading to the precipitation of these protein impurities. So phenol here working as a denaturing agent for proteins. Then we have chloroform. Chloroform increases the efficiency of the phenol for protein denaturation, and it also allows proper separation of the two phases, the organic and aqueous phase. We all know that this phenol is a kind of non-polar solution or non-polar solvent, and it has the higher density than that of the water. So it will settle at the bottom, okay, because of its higher density. But the two phases didn't get just separated the way we want them to be. So by adding chloroform, these two phases can be separated properly, right? Then chloroform also denatures the lipids as well. Then we have isoamyl alcohol. When we will discuss the different steps related to the functioning of the phenol chloroform DNA extraction method, there we will be discussing about this isoamyl alcohol in detail, that how it is going to work there. For now, you should know that when we use phenol and chloroform for the denaturation of the proteins, and when those proteins get denatured and they are settled in the interface of the two phases, that is the aqueous and the organic phase, foaming occurs. And to remove that foam from the tube, we require this isoamyl alcohol, because it prevents the emulsification of the solution. It reduces the foaming between the interface of the aqueous and organic phase. This you will be understanding when we will discuss the steps. Okay, so these are the functions of all the chemicals we are going to utilize in the phenol chloroform DNA extraction method. Moving towards the steps involved in the method. So here you can see we have an Eppendorf tube. Okay, this Eppendorf tube contains your bacterial culture. Specifically, I am telling you about the extraction of DNA from bacterial cells. Okay, so we will be focusing on that, and you can change the protocol depending upon what kind of cell you are using. So after I'm done with the bacterial cells, I will be telling you that how can you extract the DNA from the plant cell as well, just by changing certain chemicals and the ratio you are using for those chemicals, right? So the concentration and the amount of the chemical depend upon what kind of cell you are utilizing for DNA extraction. So here we are using the bacterial cells. Okay, so first of all, we will take the bacterial culture in a 2 ml Eppendorf tube and we will centrifuge it. We have centrifuged it at 14,000 rpm for 10 minutes, and when we centrifuge it, we will get two substances. First one is the upper layer, two layers you can say. First layer is your supernatant. Okay, this upper portion is your supernatant, and the settled layer is the pellet. This pellet contains all the cells, the bacterial cells. We don't require this supernatant right now because this is only the media. Okay, so we require these cells, the bacterial cells. We will remove this supernatant and discard it, and we will resuspend this pellet, the bacterial cells, into the TE buffer. Now this TE buffer is the tris EDTA buffer. Okay, you can also use lysis buffer here so that you can remove the walls, the cell wall and the cell membrane which is present on the bacterial cell. So for removing that, we require the lysis buffer, but we are using different chemicals in a stepwise manner so that you will be able to understand the function of each and every chemical we are using. So first of all, we will resuspend these cells in the TE buffer tris will provide the pH to the solution, will maintain the pH of the solution, and also helps in the lysis of the cell membrane. And EDTA also, it will degrade the DNAs and also helps in the lysis of the cell membrane. Okay, then we add SDS and mix gently. Still certain membrane structures are intact, so to dissolve them, to means degrade them, we require this SDS. We use this detergent. This will disintegrate the cell membrane and also certain proteins. Okay, then we will incubate it for 30 minutes at 55 degrees Celsius in hot water bath. So we will be incubating it for 30 minutes in hot water bath. After the incubation is done, we will add the phenol chloroform isomyl alcohol in the ratio of 25:24:1. So in this ratio, we will be adding the phenol chloroform and isomyl in the same tube containing our cells along with the lysis buffer. You can say that. Right? Now what happens? We have to mix it properly by inverting the tube. Okay? Gently we have to do that, not vigorously. So when we invert the tube gently and mix the solution gently, we will see that the homogeneous milky solution after some time appears in the tube. When this solution turns milky, what you have to do? Now you again have to centrifuge it for 10 minutes at 14,000 rpm. Right? When you centrifuge it, you will get 3 different layers in the Eppendorf tube. The first layer, the uppermost layer, is the aqueous layer. The interface means the layer which is forming between the aqueous and the organic layer. In the middle of those two solvents, aqueous and organic solvents, that layer which we called interface layer, and this interface layer consists of the protein molecules, the precipitated protein, the denatured protein. And the third layer, here you can see the last layer, this is your organic layer. Okay, so these 3 layers will be formed after you centrifuge the tube. Now what you have to do, we don't require the bottom 2 layers, that is the organic layer and the interface layer which has the protein. Now I have told you that isoamyl has a proper function here. Now what is the function? When we centrifuge this tube, if we don't use isoamyl alcohol, this middle layer, that is your precipitated protein, in this layer foaming occurs. Okay, and because of this foam, when we isolate this aqueous layer here, this aqueous layer will not be properly isolated, means you cannot remove that layer properly because of the foaming. It will hinder with that. Okay, so to reduce or remove that foam, we require the isoamyl alcohol. Right? So we use isoamyl alcohol so that we can remove the foam which occurs in the tube. Right? So you have 3 layers now. The upper aqueous layer, the center one, that is the precipitated protein, the interface layer, and the last layer, that is the organic layer. So now we don't require the organic layer and the interface layer containing the impurities, the debris, the proteins. We don't require them. Okay, what we require is the aqueous layer. This aqueous layer contains your nucleic acid, right? So we will remove this aqueous layer here in next tube, in a fresh tube. Everything should be sterile. Okay, everything you are using here is sterile. It should not be contaminated. So we will be using a sterile tube here, and we will be adding this aqueous layer in that particular tube. And after we add this aqueous layer, we will add the salt, that is the sodium acetate. You can also use NaCl, that is sodium chloride here, right? So this is how we are going to precipitate the DNA with the help of this salt. Now if you don't want to use salt, but salt obviously is going to give you better precipitation of the DNA, but if you don't want to use the salt for precipitation, you can also use chilled isopropanol. It will be having the same function as the salts. It will also help in the precipitation of the DNA. So either of this you can use, but remember that when you are using salt, don't use isopropanol. Propanol. I will tell you that why you shouldn't be using isopropanol along with salt. Though you can use, but it is not preferred. Okay, the basic means the thing is right. We should not use isopropanol with salts, because isopropanol will not remove the salt completely from the DNA when we wash it with ethanol further. Okay, so we should not use isopropanol with the salts, because ultimately when we precipitate, we have already precipitated the DNA. When we wash the DNA, what happens, this salt, if we don't remove the salt will hinder with the applications like PCR. So we don't want that. That is why we have to remove the salt from the DNA completely, and that is why we use ethanol for the washing. Okay, so when we wash this DNA, the salt will be removed, but when we use isopropanol, the isopropanol won't let this salt to be removed completely from the DNA, and at last when we use it for PCR or other application, that salt is going to hinder with that. Okay, so here you can see, you can use the salt for precipitation, or you can use the isopropanol, chilled isopropanol you can use. Then we will incubate it for 30 minutes at 0 to 4 degrees Celsius, and after the incubation, what we will do, we'll again do the centrifugation at 10,000 rpm for 10 minutes. We will discard the supernatant. Okay, the supernatant will be discarded, and then we will wash the pellet with 80% of the ethanol. Okay, and when we wash the pellet with 80% of ethanol, most of the salts will be removed and water will be removed from the DNA. Then again we will centrifuge it, right? We will get the pellet, we will discard the supernatant, and we will again wash the pellet with 70% of ethanol to remove the remaining salts. Okay, then we will again centrifuge it, discard the supernatant, and when we'll get the pellet, we will dry that pellet. When the pellet will be dried, we will resuspend that pellet in the TE buffer to maintain its integrity. Okay, to preserve the DNA. We can preserve the DNA in the TE buffer, because obviously that TE buffer is going to maintain the pH of the DNA, for the DNA basically, and it won't let the DNA degrade. So this is what the method of phenol chloroform DNA extraction, and this is how we are going to isolate the DNA. Now 2 points which I haven't completed here in detail is your precipitation method, that why alcohol is used, why ethanol is used. Though I have told you in a brief way that why we use ethanol for the precipitation in our last lesson, but I want to conclude this part in a little bit better way, so that you will get a depth about this thing related to the precipitation. So I'll be conducting a whole lesson on this, about the precipitation of DNA with the help of ethanol or other alcohols we are using, or salts, whatever we are using there. along with the quantification of the DNA, that how we do the quantification, what are the parameters we should be keeping in our mind while quantifying the DNA. Okay, so generally quantification is done with the help of spectrophotometric method. Okay, that is how we do the quantification, that is how we used to know that how much amount of DNA we have isolated. So all these things I'll be completing in another lesson that I'm going to prepare after this. So stay tuned for that, and do like the video if you like it, and do subscribe to my channel if you are new. Thank you so much. Bye bye.